Hi, good day, everyone. My name is Nacho Sanguinetti, and I'm an incoming assistant professor for the University of Pennsylvania in the US. And currently, I'm a HFSP postdoctoral fellow at Harvard University working with Hopi Hoekstra and Nao Uchida. So, today I'm going to talk to you about flexible minds, how I plan to study this going from play to wild cognition. And this is a program that has gone throughout my career and through different universities that you see depicted here. So we know a lot about the social behavior of animals from ethology, and we have learned a lot about agonistic behaviors between different conspecifics, mating rituals. We know how animals take care of their young, how they socialize with each other, and also like how they behave as collectives. But from all of this, we've mostly been restricted in studying stereotype behaviors that are very like repeated averagely in different conspecifics. But there's still a big gap to explain, and the gap is the flexibility of animal behaviors. And we've been exposed to like a myriad of videos in the internet of incredible behaviors from animals. You can see a big dog letting itself be pinned by a small dog, a turtle helping another turtle turn around, a bull letting being pushed by a goat, a, an orangutan reacting to a magic trick, and a dog saving another dog from a pool. So these are incredible flexible behaviors that just happen to happen when they happen. So the big question that I ask myself sometimes is like, how do we find this flexibility? How and where can we find this flexibility to try to explain it from a neural perspective? So one place where we find this flexibility is in social interaction. When animals interact with another being that has agency, then a lot of interesting things happen. The other place where we find this flexibility is in environmental interaction, when animals need to interact with a changing environment, whether that is changing through the seasons or changing in a very quick nature after like a disaster or in just when it rains or when it gets cold. So in that vein, we can think of interactions as the alignment of affordances. And I want to define this word for you. So, the affordance is a quality or property of an object, seen in a very general way, that defines its possible uses or makes clear how it can or should be used. A good example for this is the difference between a button, a switch, and a knob. A button has the affordance of being pushed. A switch has the affordance of being flipped, and a knob has the affordance of being rotated. And generally, we wouldn't try to confuse the affordances of these three different things because they have like a different way of interacting with them. So this concept that came from James Jerome Gibson can be applied to many other things. For example, to social behaviors. He said all social behaviors depend on the perceiving of what another person or other persons afford. So what we can do for others. A good example for this is like when you're going to greet somebody, you need to try to figure out what kind of greeting you're going to do. So it's not the same to give a handshake that to give a fist bump. So like different situations and different people might require and might have different affordances in how we interact with them. So there's people that you need to send a couple of emails until they respond. There's people that gonna respond to your email the first time, so better get it right the first try. So this can also be extended to other things like how we interact with the environment. Different environments afford for different things. So in this talk, I'm going to talk to you about two main things. So I'm going to talk to you about a behavior that is all about learning interaction. This is play behavior. And I'm also going to talk to you about a direction, an approach to studying the brain under unconstrained interaction. And I'll label this as reverse physiology. So first of all, let's talk about play behavior. This quote comes from the book Homo Ludens, written in 1938 by John Huizinga. And the book starts by saying play is older than culture. For culture, however inadequately defined, always presupposes human society, and animals have not waited for man to teach them their playing. And this is true. We see play as a behavior that appears across taxa, not only in mammals, but also in other kinds of vertebrates and even possibly in invertebrates. So, what is play? So, play can be defined as three different types of behaviors. So, play can be like playing with your own body. This is referred to sometimes as locomotive play. So, locomotive play is how animals interact with their own body and figure out the affordances or the things, the ways of interacting with their own body, and sometimes to error. Another way of playing is to interact with objects. So, for example, here is a horse interacting with a ball. So the horse needs to learn the different ways in which it can interact with the ball in the situation. So, and a final way of interacting with things or play behavior is what is called social play. And in this case, you'll see a lemur interacting with a deer. And so the lemur figures out that it can grab these knobs on the head of the deer, and the deer can figure out that it can push the lemur into the air, and the lemur will fall okay. So these are ways in which play is all about learning how to interact with oneself, with the environment, and with other beings in that environment. So, but humans uh, are very interesting in relation to play because, first of all, humans play a lot, and we all know this by experience. And they have different forms of play. They have very primary forms of play, and but they also have developed different types of play that have, in some cases, very set rules, or in some cases there's different roles. You can be a goalkeeper or you can be a striker, or in some cases the game is played by using different effectors, not just the whole body. And we know that play is an incredibly important thing for humans. It's recognized by the UN Charter as a right of the child, and it's important for the development of social and cognitive skills of the child, and it's also important for the development of cognitive and social skills of animals. So when I was doing my PhD in Berlin in the lab of Michael Brecht, we went into trying to figure out if we could teach an animal, in this case a rat, how to play a more sophisticated game that include roles and rules. So in this game, we try to teach the rat how to play hide and seek. In one case, the rat will try to search the experimenter, find it, and the only reward of this interaction and the way of teaching the rat would be to interact with it and to play with it and to tickle it. And the rat would also learn how to hide. So let's see how this looks. So this is how seeking looks like. So Annika here, our master student, puts the rat in the starting cage, then goes hiding, and then opens remotely the cage. And here I'm going to just show you, for time's sake, just one example sped up 4 times, and you'll see how quickly the rat here finds Annika. And there will be afterwards an interaction between Annika that is telling the rat that, you found me. And they will have this positive interaction with tickling and hand chasing. And so let me quickly jump to how animals can hide. So here we train rats to, like, if Annika did not close the starting box, they would know that Annika is still there. So then it's their turn to hide. And then you can see the rat will jump out, go find a hiding spot. Now, Annika here will pretend to not know exactly where the rat is for a little bit. And you can see even, like, at some point the rat will peep out of the box. There you go. And then Annika comes back, and then they have the same kind of interaction. So we have this beautiful behavior that I won't go too much into detail. You can see it. It's published since 2019 in Science. But we wanted to study this amazing behavior and record what happens in the brain during this behavior. And we did that in our original paper, but we wanted to go further. We wanted to see, like, in such an unrestricted behavior where the animal is capable of like so much interaction, we wanted to see if we could learn things from the brain directly that were not like biased by our assumptions and as experimenters. And so I want to define reverse physiology and say that there's two types of ways to trying to like understand the brain, and I think both are complementary and have their roles. One is to try to align brain activity with respect to behavior. So we know a certain behavior and we align this brain activity. This is traditional way of doing some neuroscience. Another way is to align whatever behavior happens when the behavior is very free and unconstrained with a certain brain activity that is very known and constricted. So with that, we set to study this with another master student, Ben Sibagi, where we used hidden Markov models to cluster in an unsupervised way to discover states of the brain of the prefrontal cortex of rats while they were playing this sophisticated game. Basically, if you want to understand what a hidden Markov model is, you can think about like the weather. The weather, let's say you're inside a casino and you cannot see the weather outside, but you can see how people come into the casino. You can see if they're wearing sunglasses, if they have umbrellas or mittens. And based off the different observations that you have, you can try to infer like what state the world outside is in terms of weather. So the same can be done with neurons. We have all this different observation of different neurons that are telling us something about the state of the brain. So we use the activity of neurons, of a maximum of 31 neurons recorded simultaneously, and after some dimensionality reduction, we use a hidden Markov model to try to find different states. And here you can see the different colors of the states in the bottom row are aligned with the different phases of the hide and seek game, when the animal is in the box or when it's, for example, interacting or jumping out. So this is just for one trial, but if we look throughout like a whole session of hide and seek, you can see first the variability of time of the different hide and seeking paradigm, but you can see the consistency of the states that appear through different moments of the game. A better way of looking at this is to now stretch all these states and align them to the phases of the different trials or attempts of hide and seek. So here we still had a trial structure, so we can see that this green state always happens when the animal is in the box and the box is closed. And we can see that this transit state always happens when the animal is being returned to the start box. So this we saw across sessions, and we managed to find that systematically, like with hidden Markov models, we could find states that related to the phases of the game activity. And very consistently, we found that the pairings between the behavior and the HMM states were significant across different sessions. And we found across different sessions different states that were referring to the behaviors of the animal. But with this idea of reverse physiology, we wanted to go further. We just didn't want to describe things that were already in the eye of the experimenter of important, like, for example, the phases of the game. We wanted to try to see if any of the states that are like found in an unsupervised way in the brain can tell us something about what the brain cares about that we didn't know to begin with. So we used reverse physiology to go from the activity of the brain and to try to create collections of videos of what was happening to the animal when the brain was in a certain state, when the prefrontal cortex was in a certain state. And this is very similar to what the difference between doing direct physiology with single cells. For example, you record a cell and you have the activity of the cell for different trials in relation to an event or a stimuli that you present, and you can have like an average like count of how many spikes a neuron does. Or you can go the reverse way and take the spikes and average what was the stimuli before the spike that produced the stimuli. This would be a way, this is called a spike triggered average, and is a way that people have been doing reverse physiology in other conditions. In our case, what we want is to do reverse physiology based on the states. So basically go from the states and then take collections of videos. So a good way to visualize this is to just make videos of a single state. So this is a single state, this purple state that was found in different sessions. And you can see that this state, you'll see different video depictions of what was happening during the state. And you'll see that in all cases is the experimenter bringing back the rat to the starting box. And you can see that if we plot it in the game itself, you can see that this state mostly happens always in the same place of the trial, which is at the end of the trial. But this we kind of knew. This was related to the phase of the game. But could we find other things? And we did. We did find states, for example, that were related to running. So this behavior, this state, like we're plotting all of the behaviors that the animal was doing when this state was active in the brain. And you can see all the trajectories of the rat in many different directions when the rat is like moving fast. On the other hand, we found a state that was particularly only active when the rat was had a wall to its right. And the important thing about this is that reverse physiology allows us to find brain behavior correlations that go beyond the experimenter bias, because we never would have started our experimental plans thinking, oh, we need to study if there's a wall to the right of the rat at all times. So reverse physiology and the brain is telling us something about what does that part of the brain care about the behavior and the natural statistics around the animals. So I want to go further and tell you a little bit about some preliminary work that I'm doing. So let me just start by mentioning this paper that was accepted in Nature, in which I don't participate, but was done by these amazing people. And in this paper that you will see soon, and you can check in bioRxiv, they did this amazing feat of like recording macaques that were socially interacting with their partner. And they also changed the context by changing what neighbor, changing if they had a neighbor or if they didn't have a neighbor, or the sex of the neighbor that was present. So in this case, they managed to record from freely socially interacting macaques, which is amazing. So they used this Utah arrays that is the same kind of recording technology that is used for humans, and logger devices to record from two monkeys that were socially interacting with their partners. So the question here is that we're trying to figure out in our preliminary work is, can we apply the same principle now from a macaque brain to the behavior of a pair of macaques? And we're doing this work. Camille and I are supervising this master student called Lucas Dumas, and he's working on studying like unsupervised discovery of state in macaque cortex during freely socially interacting in a no trial setting. So basically, this behavior doesn't have any trials. Hide and seek had trials. In this case, the macaques are free to interact as they wish. And we already have found that we can apply hidden Markov models to a whole recording of, like you'll see here in the time axis, 8,000 seconds, and find different likely states that you will find here for the activity of the brain. And you can see that there's some relation with the behaviors and the blocks that were tagged by the experimenter when the experimenter was doing the experiment and tagging the behavior of the animals. So there is some coincidence between the switching of the states that we find naively and unsupervisedly in the brain and the switching of behaviors. This is a more shorter time window, and you can see again these switches in states that correspond to these switches in behavior and this coincidence sometimes between the states and the behavior. And so we find, for example, that if we use mutual information, which is a way of seeing how much shared information there is between the behaviors tagged by the experimenter and the states that the HMN finds, we find that the inferred mutual information is much higher than a random null distribution. And we also find that there are pairings between different behaviors that the animals were tagged to be doing and some of the states that were recorded, that were found with the hidden Markov model. So we're advancing into this idea of like no trial, complete freedom, full interaction behavior. But now the question is, how do we add the second layer? How do we add the environment? And how do we take, so when I did my PhD, we went from having a simple maze to have the animal interacting with a human in a very complex, very free environment by using this wire-free technology. So in the future, what we want is to have animals in their own environment and record them in nature and doing their thing and completely free to pursue their own goals, the goals that they evolved to pursue. So to do that, we need to pick the animal and the environment together. And I want to present you here this animal called the agouti, which is Dasyprocta punctata, which I believe solves a lot of the challenges that rats and mice models have. And it does it by being big. It's 3 to 6 kilograms, so it's very useful for our current technology for recording the brain. It's diurnal, which makes it very easy to see and very easy to spot and to track its behavior. And it's also non-fossorial. It doesn't burrow and spend most of the time on the ground. It doesn't fly and go away from your experiment. It's non-arboreal, so you don't need to find it in a tree. And it has this beautiful brain that is larger than the brain of rats and almost the size of the brain of a macaque. And so how do you start a new animal model? So you have to meet it, and you have to meet it where it lives. So I've been working in Panama with collaborators here like Dumas Galvez, both using camera traps and trying to interact with these animals that, as you can see, are very present in the rainforest. They live all throughout the rainforest from Mexico to Argentina, and they are very present and they're amenable to humans. And I've also met these agoutis at the zoo and interacted with them, giving them nuts and seeing their natural behaviors, which are very interesting, which is, for example, the hoarding of seeds in different locations. So here you will see this agouti make a little hole and put the seed that is just now trying with its mouth, and it will make a little hole and dig it and put it there, and just cover the hole to keep it and safeguard it for when it needs it in the future. And here you can see how amenable these animals are to humans. This is an agouti taking a piece of food from my hand. Is a little bit skeptical, but at some point it will just come for its treat and take this piece of food. And so this is a really interesting animal model that will allow us to study these flexible ethograms in an organismal way, in a way that is related to the ecology of the animal, the evolution of the animal. So here is some examples of some of my field work where you can see some of the great interesting ethograms that agoutis do. So for example, here you will see a scatter hoarding ethogram. The agoutis transport food. You can see it in its mouth. Then it digs a hole. Then it places the food inside the hole. Then it covers the hole. Then not only it covers the hole, but it finds a leaf nearby and puts the leaf on top of its cover. And we still don't know exactly what is the purpose of this leaf, if it's to cover the cache or to signal where the cache is. And they also have these very interesting behaviors of treating fruits in a very different way. So different fruits might be more prone to be transported to a safe area, And some fruits are more prone to be peeled, for example. So you can see here an agouti peeling a banana and peeling an orange. And this is what we found afterwards, the remains of the banana peel and the orange peel. So, and not only that, but agoutis also interact socially and have different ways of signaling interaction with each other. So, for example, here are agoutis competing for food and doing a vocalization that we as humans can hear, which is also a very practical thing to study them. And here is another form of signaling that they use, which is visual signaling, by having piloerection of their hair in the rump, is a way of signaling in an agonistic encounter. So how do you do neuroscience in this case? So you develop new tech. So if you have a big skull, you can have a big device, and a lot of the technologies that are currently existing could really work to study the brains of these animals in a much more natural environment. So we're developing a universal logger that will have like a state-of-the-art wire-free multi-area recording with 512 channels, that will have an onboard camera and microphone so that we can see behavior from the perspective of the animal itself, that will have accelerometers to measure the behavior of the animal, and that will allow us to record with freely interacting animals in their environment for up to 8 hours. This will allow us to have multi-animal social behavior, allocentric and egocentric behavioral recordings, and we will be able to do reverse physiology to identify multi-area brain states in a condition where the animal is in its actual environment, with the actual bioacoustic statistics of the environment, with the actual visual statistics of the environment, and try to figure out what are the things that are making flexible behaviors and flexible ethograms and decision making in this animal. So this is a way that will allow us to synchronize brains with the environment and the wild and different multimodal behavior that these animals do. So for broad perspectives, today I told you about reverse physiology, and I think this is like an important thing that we need to start doing to reduce our experimental bias, to study what the brain actually is doing, or what different parts of the brain are doing and how they're communicating with each other. This doesn't mean, however, that we don't need to do very controlled experiments, and I want to clarify that a lot. I think this should be a back and forth. There should be a back and forth between creating new hypotheses and then using more controlled experiments to test those hypotheses. And the second thing is that I think we're trending to this direction of like being able to not only increase the social environment of the animal, but also to study animals that are in their natural environment. And I hope it becomes clear that this is a possibility for the future of the field that will get us closer to understanding how animals behave in a way that is related to how they evolved to behave, and how the brain is working and how the brain evolved to work. So with that, I would like to thank you and say that I'm starting my lab in January 2025, and I'm recruiting for the University of Pennsylvania. Thank you so much. Have a good day.